Welcome to Summarize Science. Have you ever wondered if it is possible to actually see what someone else is thinking? Researchers have recently developed a breakthrough technology called the Structure-Guided Diffusion Model, or SGDM. This system is designed to reconstruct visual images directly from a person's brainwaves, specifically using EEG signals. Historically, decoding EEG has been incredibly difficult because the data is full of noise and lacks fine detail. Previous attempts usually only resulted in blurry shapes or simple category labels like bird or tree. The researchers noted that these older models struggled to bridge the semantic gap between raw brain signals and complex thoughts. To solve this, SGDM uses a two-stage process that focuses on the underlying structure of what is being seen. First, it identifies the skeleton or geometric layout of an object within the brain's activity. It uses a specialized encoder to align brainwaves with a unified semantic space of images and text. Then, a structure prediction module creates a coarse map of the object's spatial relationships. This map is used to guide a powerful diffusion model, the same kind of AI used to generate realistic digital art. One of the most interesting parts of the study involved using tangrams, which are abstract geometric puzzles. These puzzles are unique because they do not have a single correct interpretation. Because tangrams are abstract, different people can look at the same shape and see totally different things. For example, one participant might see a seated person, while another sees a glass trophy in the same arrangement of blocks. The model was successfully able to capture these subjective differences, reconstructing the unique mental image for each individual. This demonstrates the model's capacity to capture participant-specific semantic representations from brain activity. When tested on natural images, the system consistently outperformed other methods in keeping the layouts accurate. It avoided the disorganized and incoherent outputs that are common in other brain-to-image technologies. The team also performed a spatiotemporal analysis to see exactly when and where these thoughts form. They found that the brain's structural encoding patterns peak between 400 and 600 milliseconds after seeing a stimulus. This is slightly later than basic visual recognition, suggesting that the brain is working hard to interpret the abstract shapes. The most useful signals came from the temporal and parietal regions of the brain, which handle high-level concepts. In summary, the researchers have found a way to use the structural bones of an image to guide AI reconstruction. This proves that EEG signals contain much more detailed structural information than previously thought. It means that we can now decode not just what someone sees, but how they specifically interpret it. This has massive implications for the future of brain-computer interfaces, or BCIs. It could eventually allow people who have lost the ability to speak to communicate through clear, vivid imagery. It could also lead to more flexible brain-to-machine communication for controlling robotic limbs or computers. This work represents a major step toward understanding the complex neural dynamics of human visual cognition. By enabling the decoding of complex visual content, science is getting closer to making mind-to-machine communication a reality. Thank you for watching this episode of Summarize Science.